Welcome folks. In this video we are going to focus on biology paper 2 for the year 2010, and now we're just going to look at section A of this paper. We have question 1. Figure 1.1 shows the structures of 4 specialized cells. So specialized cells are cells with a specific function. Now we have these 4 cells, and whenever you are studying biology, make sure you study with the diagrams, and your focus should just be on it. You should be able to identify a given question or a given structure, the name of that structure, based on how the structure is. Now let's look at the first question. So the first question, A1, using the letters of the cells, identify animal cells and name them. So the animal cells are just irregular in shape. So from these structures we have cell B and cell C. Then the name, cell B, we have a red blood cell. Then for cell C, we have a nerve cell. To specify, this is a motor neuron. Okay, we move on to question 2. For each named cell in A1, state one of its characteristic features. So the characteristic feature is what helps that particular cell to carry out its function. So for the first one, the red blood cell, we can say it has a biconcave shape. To increase a large surface area. Surface area of absorption, because we know that if you look at the function of the red blood cell, it carries oxygen, and for oxygen to diffuse from the lungs into the red blood cells, we need a large surface area. Then the other feature that we can also assign to red blood cell is the absence of the nucleus. Then if the question comes, what is the importance of not having a nucleus under red blood cells? It is to create a room for hemoglobin. Then feature in animal cell 2, which is we are looking at a nerve cell, which is a motor neuron. We can say presence of dendrites. So dendrites, for dendrites, the function is to receive the electrical impulses from the neighboring neuron. And apart from the presence of dendrites, we can also give the presence of the axon, which act as the passage for these impulses within a cell. Then also presence of we have Marian sheaths, which act as an insulator. We move on to question B. Under question B, for cells A and D in figure 1.1, name the substance found in the cell which enables it to perform its specialized function. So let's go back to cell A. So based on the structure, cell A is a phloem. Then it has the presence of companion cell. The function of the companion cell provides energy needed for the food to be transported. Then for cell D, this can be a palisade. So palisade is high concentration of chloroplast, and this is where the process of photosynthesis is high due to the high concentration of chloroplast. So for cell A, we can substance, we'll just say companion cell. Functioning to provide energy. Then for cell D, substance, we can say chloroplast. Then function, to trap sunlight needed for the process of photosynthesis. Then we move on to question E. See, figure 1.2 shows an experiment on osmosis. Okay, so based on the key, the smaller particles indicate the water molecules, then the larger particles indicate the sugar molecules. So we have solution E and solution F. Now for us to deal with osmosis, first we need to look at the solution that was highly concentrated. Then from there we need to look at where the water will move from. So we can see that from solution F there is a lot of sugar molecules. So this one is highly concentrated. Then this one is less concentrated. Now for highly concentrated, it means there is a lot of sugar molecules on solution F, but there is less water molecules. So where there is too much of sugar particles or salt particles, it means that there is a lot of water molecules. I mean, there is less water molecules. So for solution here we are saying there is more water molecules, and during the process of osmosis, the water molecules, they will move from where there is too much of water molecules to a solution where there is less of that. So let's go to question Roman number 1. From which solution in figure 1.2 will there be flow of water molecules by osmosis? So water molecules will move, we'll say, from solution E to solution F. So what of Roman number 2? Under Roman number 2, when will the flow of water molecules across the membrane stop by osmosis? So when the two solutions reach equal concentration. So whenever two solutions reaches at a point where the concentration are equal, the process of osmosis ceases to take place. Then for Roman number 3, suggests one reason why only water molecules can move across this membrane. The first one, it is selective, so it only allows water molecules to pass through, but we'll say it has small pores which allows only water molecules to pass. Okay, so we move on to question 2. Figure 2.1 shows the human digestive system, and if you look at the digestion, it's just the process by which large molecules of food are broken down into simpler ones. So we have esophagus, stomach, structure V, W, and X. So we have question A. From figure 2.1, select the letter where egestion occurs. So egestion is defined as the removal of undigested food. So this occurs through the anus. So in our case, the anus is letter X. So we put X here. Then for Roman number 2, pancreatic juice is formed. Pancreatic juice is the juice that contains enzymes that completes the digestion in the small intestine. So this one is produced by the pancreas, which is structure V. So we put letter V here. Then villi are present. Villi are present in the small intestines. So this is where the small intestines are. So we have W. Then we move on to question B. The stomach produces hydrochloric acid. State two functions of this acid in the stomach. The first one, to kill bacteria eaten with food. Then the first one we can say to activate inactive pepsinogen into active pepsin. Then we can also say the other function can be to provide the optimum pH for the enzymes to occur. Remember, the digestion that starts in the stomach is the digestion for the protein. So the enzymes that works in the stomach must make sure that the environment, the medium, is acidic. So this acid, make sure that it provides that acidic medium. Then for question C, describe the roles of the liver in number one, digestion. So under digestion, we'll say it produces a bile salt which breaks fats into smaller molecules by the process called emulsification. Then for assimilation, we are looking at the use of digested food. So, but for this one, we look at it converts excess glucose into its storage form known as glycogen by the process known as glycolysis, but let's not look into that. So the conversion of excess glucose into glycogen is one of the assimilation of the liver. We can also say that after digestion, the food must first pass through the liver before it is distributed to other parts of the body. And for the liver to do its work, it also needs some of the food. So the cells that forms the liver must also use some of the food being absorbed or after digestion. We have question 3. Figure 3.1 shows a longitudinal section through a bean seed. Okay, so the first question here is identify the parts labeled I and J. Remember, a bean seed is a dicot seed. So the outer part, which is our I, is tester. Then our J is cotyledon. Okay, we move on to describe the role of the micropyle and structure J. So micropyle, it acts as a passage for water and gases that goes in and out of the cell. Not of a cell, but of a seed. Then structure J, we said, is cotyledon. The function, it stores nutrients required by by the embryo during germination. Because for a seed to germinate, NH must be there. Then for question C, figure 3.2 shows the position of the plumule after exposure of a seedling to one-sided light for 5 days. So now the question is, what happens whenever a seedling or a part of a plant is exposed to one-sided light? So there is what we call the auxins, which is the hormones. Then the auxins, these are hormones that are responsible for growth. Now, if this is the point where the light rays are reaching this plumule here, the auxins will move to this region where there is dark. So this is what happens. So the auxins moves to a dark region, and whenever they move to this region, they will cause the cells at this region to undergo elongation. And we know that elongation has to do with the growth of cells. So whenever the cells grows in size, then that particular area will undergo a rapid growth. So that is the point. So to summarize, what I'm saying is that the point at which the light rays are reaching a plant, at that point the auxins, hormones, must move to a point where there is no light. Then the point where the auxins are concentrated, they will cause a rapid growth of the cells through the process of elongations. Thus we have the response of plants to a certain stimulus. So say this will cause the auxins to move to a dark region, and cause that region a rapid growth. Then for Roman number 2, what is the benefit of this effect to the seedling? So this effect causes the seedling to respond toward or away from a stimulus. So you know that if it responds toward a stimulus, that is known as a positive. If it is away, we have a negative. So that's the point here. So we move on to question 4. Question 4, figure 4.1 shows some components found in an ecosystem. So we have component A, B, and C. Then let's look at the first question. Which of the above component corresponds to a population? So a population is just a group of living organisms of the same species, and as long as those species can interbreed. So from these 3 components, component B qualifies. So we have component B. Then Roman number 2, a community. So a community is defined as a group of different populations. So we can see that component C has different populations. So we have component C. B1, which of the above components contains abiotics factors? So according to ecology, abiotics refers to non-living organisms. Then biotics to living organisms. So from the from this component, river is abiotic, rock is also abiotic. So say river and rock. Then which of the above component? Here we just have to state the component. So we will not give this. So the component is just component A. Then from the components, give one example. We said we can just say river. Then construct a food chain using organisms in component A in figure 4.1. So the food chain always begins with a producer. So we'll say we have producer, primary consumer. Then after primary consumer, we have secondary consumer. So just use the 3 since we are not told on how many trophic levels we are supposed to use. So let's go back. The producers should be green plants. In our case, we can use grass or trees. So let's say trees. Then under primary consumers, we are expected to have herbivores, animals that depends on plants only. So we have emperors or zebra. So let's use emperor. Then the secondary should be carnivora, animals that depends on other animals for flesh only. So we may have lions here. Okay, we move on to question C. Figure 4.2 shows untreated industrial effluent being discharged into a river. So the thing is about the discharge of this into a river. Then Roman number 1, what type of pollution is shown in figure 4.2? So if you can see, there is the discharge of smoke into the atmosphere and also of these chemicals into the river. So we have 2 types of pollutions, but to focus on water because we are taught from the question the discharge of these unwanted substances into the river. So say water pollution. Then suggest a pollutant which can be found in the industrial effluent. So we can say nitrogenous waste product. Okay, we move on to what measures can be taken to reduce this type of pollution. So say by treating the waste chemicals before being discharged into a river. Then we go to the last question. So under the last question, figure 5.1 shows the inheritance of hemophilia in a family. Then the key, a box that is shaded. We have male hemophilic, shaded circle, female hemophilic, and shaded box, normal male, then normal female. Now first let's see if we can look at some. We'll first begin by giving them some genotypes for a man. So when a man is 100% okay, this is the genotype. If the man is sick, this is the genotype. So for a man, there is only 2 options. We have sick or okay. Now for a woman, the case is different. A woman who is 100% okay will have capital letters HH. Then a woman who is a carrier must have 1 capital letter and 1 small letter. Then for a woman who is sick, we have small letters. we need these genotypes to help us to identify. Now, if you look at the given question, this man is sick, but the woman is okay. Now, this kind of okay can be 100% normal or can be a carrier. Now, how can we tell that the genotype that we can use for the parent, which is 1 here, we are going to use the carrier or for normal? We need to look at the offsprings. Now, let me give you a condition. If among the offspring there is at least one girl child who is affected, and the husband is sick, then the woman must be a carrier. That's a point. Why? Because if the woman is 100% okay, then the man is sick, they cannot have a child who is hemophilic. They can only have a son. Yes, the son who is hemophilic, that is possible. But for a daughter to be hemophilic, we need at least one small letter from each parent. So these two, the mother is a carrier, then the father is sick. Then the question is, taking the alleles for hemophilia to be XH, what is the genotype of offspring 3? So offspring 3, we can see that this is a woman and this woman is sick, and we said for this one to be sick we just need small letters. Then offspring 7, the man is okay, is XY. You just put the letter X, you attach to the allele X. Now we move on to question B. Offspring 4 married a person with similar genotype to offspring 7. So 7 we have. Now for offspring 4, this one, we don't know if this is a carrier 100% okay. So we first have to look at this. Now let's go back to the parent. So we said for a man is known, is sick for the parent. Then for the woman, we said we need to have a carrier. So let's look at what we can have. So this one, so we can see that this girl child is a carrier. Then this girl child is sick. Then this boy child is okay. This boy child is sick. So for the chances of these two parents is that they only have a chance to have a daughter who is a carrier or sick. They cannot produce a child who is, a girl child who is 100% okay. So that's a point. So this is a patient, which is this one. Then this is a patient, which is also this one. Now for this one must be a carrier, which is this one. Even this one must be a carrier, which is this one. Because we are saying they do not have a chance to have a girl child who is 100%. Okay, so we are dealing with a carrier here. So let's have a genetic diagram. So we'll start first by saying parental phenotype. So say the man. So we have the woman. So for the man which is 7, we are saying normal. Then for the woman, we'll say normal carrier. Then parental genotype. For the man, which is, it is compared to offspring 7, so we are using this. So we'll say X, H, Y. Then carrier is non-X, H, X small letter, H. Then we'll move on to parental gametes. You just have to separate these. Okay, after that we move on to random fertilization. Here, so there is a chance of these having a girl child who is 100% okay. Another chance of having a girl child who is a carrier. One chance of having a boy child who is 100% okay. Then another chance to have a son who is hemophilic. So say genotype of F1. Then we move on to question Roman number 2. So what is the probability of them having a normal child? So a normal child, this is normal, normal, normal. So for this time, normal, we don't care whether carrier under women is still normal. So say probability of normal is 3 over the total is 4, which is, if this is a chance, is 75%. Then Roman number 2, what is the probability of them having a child who is a carrier? So for probability of carrier will be, carrier is only 1 over 4. Then if we want to take this one as a pure normal, then pure normal, it's 2 over 4, which is half. So this is what we can share for this video. Thanks for watching.